Bismillahirrahmanirrahim. Today we are going to discuss neuromuscular junction, and first we'll discuss structure of neuromuscular junction, that what structure are included in the neuromuscular junction. So neuromuscular junction contain motor neuron, muscle fiber, synaptic cleft, and axonal terminal. The muscle fiber are consist of skeletal muscle, smooth muscle, and cardiac muscle fibers. And axon terminal contain acetylcholine, vesicle, mitochondria, and transport channel. And the muscle also contain transport channels. So this is the structure of neuromuscular junction. Here you can see the axon terminal, which contain the mitochondria, the acetylcholine vesicle, the acetylcholine, and here we have the muscle fiber. This fiber, between the fiber and the nerve terminal or the axon terminal, there is a space which is called synaptic cleft. This junction is actually between the nerve and the muscle fiber, and the nerve is actually a motor nerve and it is a myelinated nerve, and at the end its axons branch and they supply multiple neuronal fibers, muscle fiber. The neuromuscular junction is a synaptic connection between the terminal end of a motor nerve and a muscle fiber. Muscles are skeletal, smooth, and cardiac muscles. What is synapse? Synapse is a close contact between two cells to share information with each other. It is the site for transmission of action potential from nerve to the muscle. Here we have a nerve, axon terminal. Here the action potential arises, and this action potential is transmitted to the nerve fiber. This traffic is one way, that the action potential come from the neuron. It is converted into a chemical signal, and then the chemical signal is converted to the electrical signal. So this is a one-way traffic from neuron to the muscle. The skeletal muscle fiber are innervated by a large myelinated nerve fiber that originate from large motor nerve in the anterior horn of the spinal cord. This is a motor nerve. You can see that this is the posterior horn and this is the anterior horn, and this originate from the anterior horn. This is a large motor nerve, and its terminal fiber, the axon terminal, supply the muscle fiber. These are the muscle fiber which are innervated by the motor neuron. Here you can see this is the dorsal motor neuron and this is the ventral motor neuron, and these fiber originated from the ventral motor neuron and then supply the muscle fiber. This neuron is myelinated and multipolar neuron. You can see this. This neuron, motor neuron, is covered by a myelin sheet, and these are the axon terminal, and a neuronal button make a junction with the muscle fibers of skeletal, cardiac, or smooth muscles. The neuromuscular junction acts as a bridge between the nervous system and the muscular system. It is a bridge, or it is a uh, connection between the central nervous system and the muscular system. It receives information from the nervous system, and that both are connected with each other. They bring information from the nervous system, and from the nervous system, information are transmitted to the muscular system. It is a microstructure through which the process of contraction is initiated or halted in the muscle by the neuron. At this point, either the muscle is stimulated or inhibited, and this inhibition and stimulation is carried out by the neuron. Any change in the neuromuscular junction can result in impaired contraction of the skeletal muscle. When there is any abnormality in the skeletal muscle at the neurojunction muscle, it can impair the contraction of the skeletal muscles. These abnormalities may be either drug, disease, and maybe toxins. We will discuss them in the last of this lecture. Each nerve fiber after entering the muscle belly. This is the muscle belly. When the nerve enters the muscle belly, normally branched and stimulate from 3 to several hundred skeletal muscle fiber. Here we can see this is the nerve. When it enters the muscle belly, it stimulates from 3 fibers to 100 skeletal muscle fibers, several hundred skeletal muscle fibers. Each nerve ending makes a junction with a muscle fiber near its midpoint. So this is a muscle fiber. So this junction, the nerve and the muscle junction, is at the midpoint of the muscle fiber. The action potential initiated in the muscle fiber by the nerve signal travel in both directions towards the muscle fiber end. When action potential arises here, arrive to the junctions by these presynaptic neuron, this action potential to the midpoint of the muscle, when it arrive, it spread in both directions, from here to here, and from the midpoint in the left direction and in the right direction. With the exception of about 2% of the muscle fiber, there is only one such junction for muscle fiber. 2% of the muscle fibers are not supplied by or they have no muscular junction. The rest 80% of the fiber have such type of neuromuscular junction. The nerve fiber form a complex of branching nerve terminal that invaginate into the surface of the muscle fiber but lie outside the muscle fiber plasma membrane. There is invagination, not actual penetration into the muscle or the muscle fiber or the muscle membrane. It makes a depression in the muscle fiber. The entire structure is called the motor end plate. This neuronal terminal, the muscle membrane, the space between the muscle fiber and the and the terminal neuron, and its sheeting. This structure is covered by the myelin sheet. So this whole structure in which neuron, synaptic cleft, muscle fiber, content of the neuron, and content of the muscle fiber. All these structures are known as motor end plate. It is covered by one or more Schwann cells that insulate it from the surrounding fluid. You can see here this neuron is covered in this nerve end nerve plate. Motor end nerve plate is covered by the Schwann cells, so it insulates. Like in electrical cable, we have a coating on the electrical cable. The same is there is a cable-like structure which is made from the Schwann cell. It covers the neuron and it insulates the neuron from the surrounding structures. Synaptic gutter and synaptic clip. The invagination membrane is called a synaptic gutter. This is the membrane, and at this point it is invaginated or depressed, and this invagination is called the synaptic gutter. And the space between the nerve terminal and the fiber membrane is called the synaptic space or synaptic clip. Here you can see that this is the neuron terminal, axon terminal, and here we have a space. This space is called synaptic space or synaptic clip. This space is 20 to 30 nanometer wide. The space between the neuron terminal and the membrane is 20 to 30 nanometer wide. At the bottom of the gutter are numerous small pores of muscle membrane called subneural clefts, which greatly increase the surface area at which the synaptic transmitter can act. You can see that this here the on the surface of membrane which make a junction with the neuron. This area on the muscle surface is folded. So this actually increase the area for the neurotransmitter. If we will stretch this area, this will be enlarged. So the folding of this membrane increase the area for neurotransmitters. In the axon terminal are many mitochondria that supply adenosine triphosphate ATP, the energy source that is used for synthesis of excitatory transmitter acetylcholine stored in the vesicle. You can see that the neuron terminal, and this neuron terminal we have mitochondria. This mitochondria contain ATP, which is adenosine triphosphate. This provides energy for the synthesis of acetylcholine and then transportation of this acetylcholine into the vesicle. The acetylcholine excite the muscle fiber membrane. When this uh, acetylcholine from the vesicle are released to the synaptic cleft, it uh, act on the muscle fiber, which have receptor for the acetylcholine, and then it excites the muscle. Now we have the uh, acetylcholine vesicle in the nerve terminal, but where they are synthesized? They are actually synthesized in the body or soma of the neuron. The synthesis of transmitter vesicle begin in the endoplastic reticulum of the nerve cell body, where the protein that make up the vesicle are synthesized. We know that here in this body we have the nucleus, and the nucleus have DNA. From DNA, protein synthesis occur by uh, translation and by protein synthesis mechanism. Protein is synthesized and then it is transported to the Golgi apparatus. From Golgi apparatus, it is loaded into a transporter. Continue as the protein migrate through the smooth endoplastic reticulum and the Golgi apparatus to eventually emerge to the transported to the nerve ending. This is loaded to the transporter protein and ultimately it reach the nerve ending. At the beginning, these vesicles, when they arrive at the nerve ending, they are empty. They are empty and thereby mechanism which we will discuss in the next slide are then loaded with the acetylcholine. Here they are synthesized here and then they reach to the nerve ending. This transportation is called past anterograde exoplasmic transport. From when these vesicles are formed, they are loaded to this protein which is known kinesin, and it is a energy dependent process. This transportation is an energy dependent process, and this energy is supplied by the kinesin protein, and ultimately they transport these vesicles from the soma to the neuron terminal. There is another theory that these vesicles, is because in long neuron the soma is far away from the neuronal terminal, so here large number of these vesicles are present. So these vesicle synthesis in such a large number is not easy. So another researcher agree that these vesicles are recycled. So the synaptic vesicle recycle take place in steps. These steps are synaptic vesicles are loaded with neurotransmitter by the active transport. I told you when these vesicles rise to the nerve terminal, they are empty and then they are loaded with the uh, acetylcholine by active transportation. Newly filled synaptic vesicles are then transported to the active zone of the synapse. Here, this is the active zone of the synapse is the membrane, the proximal membrane of the nerve ending. So, when they are loaded with the acetylcholine, they are transported to the nerve membrane, the presynaptic axonal membrane. Newly filled synaptic vesicles are then transported to the active zone of the synapse, a process known as tethering. They are tied with a protein, and these protein pull these neurons here to the active zone. The synaptic vesicles are then docked to the presynaptic membrane. They are then placed or docked or loaded on the synaptic membrane. Synaptic vesicle status become fusion competent after a priming step awaiting calcium signal. These vesicle and also the acetylcholine is sensitive to the presence of calcium. When calcium enters into the neuronal end by stimulation, then they are now mature or competent, and by this entry of the calcium into the neuronal end cause fusion fusing of the or merging of the vesicle and the membrane, and then the vesicle actually uh, release its neurotransmitter into the synaptic cleft. After this release, uh, the calcium signal trigger fusion process, and few synaptic vesicles are then coated with clethrine and are prepared for endocytosis. In recycling, one mechanism is that these vesicles are synthesized in the soma. The other mechanism is that when these vesicles are emptied, they are coated by lethrine, and then it is endocytosed, and then it is recycled. Endocytosed synaptic vesicle then shed the clethrine coat and are acidified via protein pump. This membrane is then removed from, when this is endocytosed, this membrane is then removed from the vesicle, and this vesicle then later fuse with the endosome. This is a structure which is present in the uh, neuronal end, which is called endosome. This fuse with the endosome, and when it is become fully synthesized, the vesicle is fully synthesized, and when it is ready, it is detached from the this endosome, and then it is recycled, reloaded with the acetylcholine, and pass through the same mechanism as we discussed in the previous lecture. Another mechanism is that these vesicles are bypass these steps from 7 to 9. These steps are actually they are bypassed, and the vesicle directly jump to the step 1. They don't the vesicle doesn't attach with the endosome, but directly uh, goes to the step 1 and reloaded with the acetylcholine, and then reuse for the production of action production of synaptic activities by releasing acetylcholine into the synaptic cleft. Now, this was the story of synthesis of uh, acetylcholine cycle. Now, here we will discuss how the acetylcholine is synthesized. Choline is actually present in most of the food, especially those food which contain protein, and when these cholines are taken up by transporter channel which are present on the neuronal membrane. And when this choline is taken inside to the neuro, pre-synaptic neuronic cytoplasm, in here we have mitochondria, and by pyroic glucose and pyruvate, this mitochondria actually produce acetate or acetyl CoA. Under the influence of an enzyme, choline acetyl transferase, when it acts on these two, acetyl CoA and choline, when this enzyme acts on it, it synthesizes acetylcholine, and then this acetylcholine is loaded by active transport to the vesicle. Now secretion, when a nerve impulse reaches the neuromuscular junction, about 125 vesicles of acetylcholine are released from the terminal into the synaptic space. This is the uh, neuromuscular junction, you can see, and this is the neuron terminal. When the action potential arises, it actually releases the acetylcholine into the synaptic cleft, and at once by one stimulus 125 vesicles are release their acetylcholine into the synaptic space or synaptic cleft. On the inside surface of the neuronal membrane are linear dense bar. You can see that here is a dense structure. This is electron. Here high concentration of electrons are present, and this area is called the dense bar. To the each side of each dense bar are voltage gated calcium channels. This is the dense bar, and you can see that there are calcium voltage gated channels. They are present here on both sides of this dense bar. When an action potential spread over the terminal, these channels open. They are now in normal condition, these channels are closed, but when action potential spread uh, here, these channels are open and allow calcium into diffuse from the synaptic space to the interior of the nerve terminal. This is the synaptic space. When action potential arrive in the nerve terminal, these channels are open and the calcium actually rush from the synaptic space into the nerve terminal cytoplasm. The calcium ion in turn are believed to exert an attractive influence on the acetylcholine vesicle, draw them to the neuronal membrane adjacent to the dense bar. Here we have the vesicle, and on the other hand, here we have the axonal membrane, the nerve membrane, which make the neuromuscular junction. This is the membrane. On the platelet, there is a receptor which is called synaptobrevin. Synaptobrevin, this is a receptor which is present on the surface of platelet, and on the surface of neural membrane another receptor is present which is called syntaxin. These two receptors, when the calcium enter into the uh, from the synaptic cleft into the cytoplasm, they by the action of they activate these two receptors, and they attach or attract to each other. These receptors then come closer to each other and they join with each other, and then this vesicle is fused with the, here you can see that these vesicles are fused with the neuronal membrane. They, by joining these, when these two receptors join each other, the vesicle and the membrane actually share their information, and by sharing this information, this vesicle is attached or fused with the membrane, and by this fusion, this is the vesicle is ruptured and it releases its neurotransmitter to the neuronal clip or neuromuscular junction clipped. These acetylcholine, there are acetylcholine gated ion channel, and they are located almost entirely near the mouth of the subneural cleft lying immediately below the dense bar area where the acetylcholine is emptied into the synaptic gutter. In front of the dense bar, this is the now the synaptic cleft, and after below the synaptic cleft is the muscle fiber. These are the fold in the muscle fiber. In front of the dense bar, there are receptors are present on the muscle fiber, and these receptors actually are they actually attach the acetylcholine. RAPSN. This is a protein and which is 43 kilodalton receptor associated, R per receptor, A per associated protein of the synapse, is a protein that in neuromuscular junction that maintain synaptic structure and result in the aggregation and localization of the acetylcholine receptor on the postsynaptic port. This protein is responsible for the maintenance of the uh, synaptic cleft and also for the localization or aggregation of the acetylcholine receptor on the membrane. This is the repair, this do the repair work of the synaptic cleft and aggregation of the channels, acetylcholine sensitive channels. Now, these receptors are present on the muscle membrane. On the muscle membrane, the receptors which are present, they are actually made of 5 protein. These proteins are 2 alpha protein and 1 each beta, delta and gamma protein. These protein molecule penetrate all the way through the membrane lying side by side in a circle to form a tubular canal. Here you can see that these are 5 protein. 2 are alpha, beta, and then delta and gamma protein. They are 2 alpha, this is gamma, and beta, delta are these are beta, delta contain 5 protein, and they are arranged in such a way that they make a barrel-like structure. In normal condition, the channel remain closed. When there is no activity in the muscle, this channel remain closed. Here you can see that the channel is closed. But when the action potential arises, here the alpha channel make receptor for the acetylcholine. The acetylcholine which is released from the neuronal end to the synaptic gutter. This is acetylcholine then attached to the alpha channels, and by attaching there are two ends on the this receptor, and these two ends are formed by the alpha uh, protein of the channel. When when the acetylcholine attached to the both of these receptor area of the uh, channel, it is activated and it become open. This causes a conformational change that open the channel. When the channel is open, sodium ion flow faster through the acetylcholine channel than any other ion for two reasons. The sodium rushing to the inside is faster than any other ions or positive ion which are present outside the membrane. There are two reasons. First, there are only two positive ions in the large concentration, sodium ion in the extracellular fluid and potassium ion in the intracellular fluid. We have two ions, and when the membrane is in resting position, there are two positive ion. One sodium is outside and potassium is inside. The open acetylcholine channel has a diameter of 0.65 nanometer, which is larger enough to allow the important positive ions sodium, potassium, and calcium to more easily through the opening. This channel is normally closed, but when by the action potential it is open and it has a diameter of 0.65 nanometer through which sodium and other calcium, potassium can easily pass. Negative ions such as chloride ion do not pass through because of strong negative charge in the mouth of the channel that repel these negative ions. You can see that this channel is negatively charged. The mouth of this channel is negatively charged, so this doesn't allow the negative charge ions to pass through this channel, like chloride, which is negatively charged. Now, as I showed in the previous slide, that outside is sodium and inside is potassium. Both are positively charged in resting condition. When the membrane is resting, the muscle resting potential is minus 80 to minus 90, and this resting potential is maintained by the potassium. This is the maximum power of the potassium which retain the resting membrane potential. So one reason is that why the sodium rush into the when there is action potential, or when there is acetylcholine are released and attached to the its channels and they are open, so the sodium rush inside and there is increase in the resting membrane potential from minus 80 to 0. And because the potassium want to maintain this negativity, so it, due to this delay, the sodium ion enter the muscle membrane or muscle cytoplasm earlier than the potassium leave the membrane. the inside of the membrane. So the negativity is converted from -80 to 0, and this create action potential. This create potential. Remember, this potential is not action potential, but it is a channel potential because the sodium is transported through this channel, acetylcholine channel, which is opened by the acetylcholine channel for the sodium, and the sodium rush through these channels into the inside of the muscle cell. Now when the action, this uh, receptor potential is produced here, or channel potential is produced here, it's propagate on both sides of the muscle, and then it produce the action potential, and this cause the contraction of the muscle. Opening the acetylcholine gate channel allow a large number of sodium ion to pour to the inside of the fiber with positive charge. This create a local positive potential change inside the muscle fiber membrane called the end plate potential. When these receptor the sodium into the muscle, this cause a potential which is called end plate potential or receptor potential. This is not action potential, but it is a receptor potential. This end plate potential initiate an action potential that spread along the muscle membrane and thus cause muscle contraction. Now, when the action potential is poor and contraction of the muscle is poor, so then the acetylcholine, which cause the contraction of the muscle, is removed from the synaptic cleft, and acetylcholine rapidly by two means. Most of the acetylcholine is destroyed by enzyme acetylcholine esterase. Here, acetylcholine esterase is present in this synaptic cleft, uh, mainly attached to spongy layer of fine connective tissue that fill the synaptic space between the presynaptic nerve terminal and the postsynaptic muscle fiber. This is the presynaptic nerve terminal, and this is the postsynaptic muscle fiber. This choline esterase is present here in this space attached to the spongy tissue, and they split the acetylcholine into acetate and choline. This is one mechanism that it is the acetylcholine is destroyed in the synaptic cleft by the acetylcholine esterase. The other mechanism is a small amount of acetylcholine diffuse out of the synaptic space and is then no longer available to act on the muscle fiber membrane. Some of the acetylcholine diffuse to the surrounding structures. Now, here, as we have discussed this, that where from the choline come and how the acetylcholine is synthesized. So, from the food, proteinish food, the choline come, and by the choline pump, it is taken into the inside of the nerve terminal, and here by the uh, this acetyl-CoA is produced by the mitochondria and by the action of CAT. CAT means choline acetyl transferase. This is an enzyme which acts on these two and bind them, and they synthesize the acetylcholine, which is then loaded to the synaptic vesicle. So here you can see that This is a mechanism which takes place at the site of the neuronal uh, at the neuromuscular junction. Here you can see this is the electrical potential or electrical current which arrive from by motor neuron to the presynaptic neuron. Here we have the vesicle which contain the acetylcholine by the mechanism which we have discussed, which we discussed in the previous slides. These vesicles are fused with the membrane and the exocytoses are release acetylcholine into the synaptic cleft. Then this acetylcholine is attached to the uh, receptor which are present on the muscle membrane, and they are taken by the muscle membrane. These channels are open, sodium rush to the muscle, and it causes the first, this is the electrical, it is converted into chemical, and the chemical is again converted into electrical potential, and then uh, the, the muscle contraction occur. Here in this animation, you can see the how the muscle. Now this was the whole story of the physiological anatomy of the neuromuscular junction. Now we will discuss safety factor for transmission at the neuromuscular junction, fatigue of the junction. Each impulse that arrives at the neuromuscular junction causes about 3 times as much as end plate potential as that required to stimulate the muscle fiber. The action potential which arises at the uh, end plate, it is 3 times greater, or it is 3 times more for the production of action potential in the muscle. So this is the safety factor. A very small stimulus, a small frequency stimulus can activate the muscle fiber, because the end plate potential responds 3 times more to a stimulus which arrive at the nerve terminal. Therefore, the normal muscular junction is said to have a high safety factor. In case when if there is any abnormality, this can save the muscle from fatigue, easy fatigue. However, stimulation of the nerve fiber at a rate greater than 100 times per second for several minutes often diminish the number of acetylcholine vesicles so much that impulses fail to pass into the muscle fiber. Like in stressful exercise, and if the impulse to the uh, neuromuscular junction is more than 100 times per second, it causes release of rupture of more vesicle and release of more acetylcholine, and with this with the same frequency or rate, the acetylcholine and the vesicles are not sensitized in the nerve ending. So in that condition, in stressful condition, when uh, more than 100 times the muscle is stimulated, or it is put under stress, like in this exercise, the muscle goes into fatigue. This is called fatigue of the muscular junction. Under normal function condition, measurable fatigue of the neuromuscular junction occur rarely, and even then only at a most exhausting level of muscle activity. This phenomena that the end plate potential is as 3 times more than the potential which arrive at the uh, presynaptic nerve ending. This prevents the fatigue of muscle in normal condition, but in a stressful condition, the muscle can or maybe uh, goes into fatigue. Now, myasthenia gravis, as we discussed early in the first slide there, or in the definition of the neuromuscular junction, that abnormality of the neuromuscular junction or diseases, toxin, drugs, they can alter the function of neuromuscular junction. So myasthenia gravis is one disease which affect the uh, function of neuromuscular junction, and this actually occur one patient in 20 births, one patient is suffering from this disease. This is actually an autoimmune disease, and antibody attack the acetylcholine gated sodium ion transport protein have been demonstrated in the blood of a patient with myasthenia gravis. These antibodies are actually detected in the or they are present in the blood of such patient, which actually destroy the acetylcholine gated sodium channels which are present on the muscle membrane, that of the 5 proteins, the alpha, beta, gamma, and delta. They destroy that protein. The end plate potential that occur in the muscle fiber are mostly too weak to stimulate the muscle fiber. If the disease is intense enough, the patient die of paralysis, particularly paralysis of the respiratory muscle. The disease usually can be ameliorated for several hours by administration of neostigmine or some other anticholinergic drug, which allow large than normal amount of acetylcholine to accumulate in the synaptic space. Within minutes, some of these paralyzed people can begin to function almost normally until a new dose of neostigmine is required a few or later. So we have less receptor for the acetylcholine. So actually acetylcholine is produced from the nerve terminal, but there are no receptor on the muscle fiber, so they are actually not able to produce action potential in the muscle fiber. When new stigmine is given, it prevents the destruction of acetylcholine. So large amount of acetylcholine is available in the synaptic cleft, and when this drug is given to a patient, he can within minute he can perform his normal work for many hours until uh, the effect of this drug is uh, subsided, and for further activity the patient need another dose. This is a patient of myasthenia gravis. Early sign of myasthenia gravis are ptosis of the eyelid or dropping of the eyelid and facial muscle. This also occur in the facial palsy, but here other symptoms are also associated. This patient become fatigue very easily. When we try to talk with this patient, he can talk for he can speak few words. After that, he stop talking. Or when food is presenting to him, he because his muscles are paralyzed, he cannot take the food, many food. So this is the difference between facial palsy and the myasthenia gravis. In myasthenia gravis, the patient fatigue very easily, and this is due to these are the acetylcholine receptor, and antibodies are produced against these receptors. They are destroyed. They, so there are less receptor on the membrane of the muscle, and the acetylcholine cannot pass to the muscle. That's why it cannot contract. Here you can see this is a normal receptor, and here acetylcholine is has rushed to the muscle fiber. But here the acetylcholine, the the receptor is damaged by the antibody, so the acetylcholine cannot pass to the muscle and cannot produce action potential. The other disease is labor-eaten myasthenic syndrome, LEMS. In this condition, actually the the calcium channels are destroyed. The calcium channels are blocked in this condition. So when action potential arises here, the calcium cannot uh, enter into the neuronal end, and when there is no calcium, these acetylcholine uh, vesicle cannot attach with the neuronal membrane and cannot release its acetylcholine. In this condition, the patient present like a myasthenia gravis. But the difference between in myasthenia gravis and Lambert-Eaton myasthenic syndrome is that if you will present food to such a patient, the the eaten Lambert myasthenic syndrome patient will uh, at first he will start slowly, but after some time he will take the food easily. And in case of myasthenia gravis, the patient take few uh, little food, but after that he refused to take or he cannot take more food. So he, with exercise, the myasthenic patient become fatigue, fatigue, fatigue easily. But in Lambert-Eaton syndrome, with exercise, the activity is improved. He is now active with exercise. Botulism, this is a poison produced by bacteria which is called Clostridium botulinum. This is produced by this bacteria, and this actually block this is the botulism patient. You can see here also the facial muscles and the uh, eyelids or drop due to the weakness. In this condition, the acetylcholines are prevented from release to the synaptic cleft. This is in botulism. The acetylcholine are these vesicles are prevented to release its neurotransmitter to the synaptic cleft, and when there will be no acetylcholine, the receptor on the muscle fiber will have no acetylcholine for stimulation, and in this case the contraction will not occur, and this leads to the paralysis of muscles. Curare poisoning, this is another uh, uh, poison which cause paralysis of muscle, and it was used in the Africans. They were used this poison on their arrows for killing for hunting the animal. They were apply this poison to the arrow, and by this arrow they were able to paralyze the animal, and in this way they was using this poison for hunting. The mechanism of action of this poison is to core the receptor, the acetylcholine receptor on the muscle fiber. Now, in this case, acetylcholine is available, but the receptors this poison actually block the receptor and cannot pass the uh, action potential to the muscle fiber, and in this way paralyze the muscle. This bacteria, this uh, Clostridium botulism, it is actually occur in this is grow in aerobic uh, condition. So the food which are stored and aerobically in can, and if it is not uh, stored properly, especially the fishes, so these toxins are produced in their food, and if somebody take this food, it cause the blockage of these receptors, and it lead to the paralysis of his muscle, and it can lead to the death of patient. This curare is also produced. The curare is obtained from a plant. This is a plant, and this is a seed from by heating, boiling and heating these seed. This poison is obtained from the seed of the curare. So this was whole about the neuromuscular junction. So in this we learned that the neuromuscular junction is a synaptic connection between the terminal end of motor neuron and the muscle fiber, which are skeletal, smooth, and cardiac muscle. Neuromuscular junctions allow all voluntary movement and ensure vital function like breathing. It is the site for the transmission of action potential from nerve to the muscle, and it is also a site for many diseases and a site of action for many pharmacological drugs. So thank you. Please subscribe the channel and press the bell icon for in-time notification, and leave your valuable comment for improvement of the channel. Thank you once again.